Okay, so next up we have Dr. Jennifer Friedman from Rady Children's in San Diego.
So thank you very much. I wanted to address quickly the parent who asked about chorea versus dystonia. I just wanted to say I've been doing movement disorders for decades, and I still have scratched my head at times. So it's not easy, and it's very, very difficult, even for people who are supposedly experts in this. So for my talk, I first want to thank the Bow Foundation for inviting me to speak here today and for supporting our efforts to try to develop an individualized patient therapy for Levi, who is somewhere here in this room today. The therapy is called an antisense oligonucleotide therapy, and I will get to what that means as I go through the talk. Because we have a mixed group here today, I am going to start fairly basic. I hope that's okay, and spare some of the details for people who want to ask questions of me later. So just starting with some background, you all know that GNAO1 is incredibly rare disorder. There's no real definition for what constitutes rare disorder, though the U.S. Orphan Drug Act defines a rare disorder as one that affects fewer than 200,000 people. And by those statistics, clearly GNAO1 is an ultra-rare or ultra-rare disorder. And as most of you know, those with ultra-rare disorders often struggle to find diagnoses, but thankfully genome sequencing has dropped in cost, and this has really accelerated the rate at which we're able to find diagnoses for patients. I don't really have any accurate number of how many patients have been diagnosed with GNAO1. A quick literature search shows that clearly there is exponential growth in interest and awareness of this condition, which is, of course, a good thing. But also, as many of you know, diagnosis really is the first step in a long journey, and though sequencing has really shortened the time to diagnosis, for many rare conditions, including GNAO, treatments have seemed far away. So some of the reasons that treatments take so long has to do with the really long and arduous process that one must go through to develop drugs. And these include the time from concept formation in the lab to implementation into clinical trials, and eventually, at least in the US, FDA approval, and elsewhere, other regulatory body approvals. And this can take immense amounts of time and money. These are some statistics from 2014 looking at costs in the US for development of a single drug. And as you can see, it's quite high. And in part, the process is so lengthy due to the need, at least historically, to treat each individual drug as a unique structure with unique impacts and unique side effects, to treat them as snowflakes, as if you will. And each of these aspects of every single drug needs to be evaluated independently, which is a very, very slow and inefficient process. But this is beginning to change. The genetic discoveries have enabled us to implement genetically targeted therapies where we can apply lessons that have been learned for therapies for one rare disorder to genetic therapies for other conditions, and thus speed up the process and improve the efficiency of the process. So utilizing just an analogy of making a tool set, it's a very inefficient process to develop tools with unique shapes for every single different purpose. A modular approach where there may be a standard base with different parts that each have a unique purpose but can be switched in and out to that single modular base is a much more efficient process. And similarly, utilizing a similar base for a drug and switching in and out different pieces that can impact different diseases is a much more efficient process for developing drugs, and one that we are able to now use with our genetic therapies. To go into a little bit more detail here, each drug has a portion that is responsible for the uptake, the distribution, and the safety, and a separate part, or sometimes intermixed part, that is responsible for the effector portion of that drug that impacts the biologic target. In most drugs, these aspects are linked and difficult to separate one from the other, and thus each drug is unique and can't easily be modified. The informational drugs, in contrast, and I'm sorry, I do have some problems with the slides, but I think you can read it. In the informational drugs, these parts are separable, and so we can use the modular approach by having a single part that is responsible for the uptake, distribution, and safety, and has been tested and established as safe and efficacious, and we can substitute in and out the portion of the drug that is responsible for the effectiveness or the biologic target. And this is the strategy that is used with the new genetic therapies, of which the antisense oligonucleotide therapies are perhaps the most mature example. And in order to help you understand how antisense oligonucleotide therapies work, I'd remind you of the process that happens for our DNA to provide the instructions for building the proteins in our body. DNA sends its message through mRNA to the machinery that produces the protein in our body. And the, sorry, we again have a little change there on my slide, but the antisense oligonucleotides interfere with that process by binding to the messenger RNA and interfering with how it works. And these ASOs can bind directly to specific genes and to specific areas of specific genes depending on what the disorder is and what the specific mutation is. And so you can see they can be very personalized, not only to a disease, but to a particular mutation, and are even further personalized to an individual patient with a particular mutation. These can work in a number of different ways. The decreased production of harmful proteins, as you see on the top, there's some white boxes covering some of the words, but they can also increase the production of beneficial proteins and modulate the RNA processing and production of modified proteins. Sorry, it's coming through now. The antisense oligonucleotide therapies need to be delivered by spinal tap or lumbar puncture in order to reach the brain as well as other parts of the nervous system. And though this is a pretty cumbersome process, there have really been some spectacular successes. The most notable of these is the development by Ionis Pharmaceuticals of nusinersen for spinal muscular atrophy, or SMA. This had been, until recently, one of the most common causes of infant mortality in the U.S., and is a disorder that's characterized by progressive motor dysfunction and eventual death. The development of nusinersen has really transformed the trajectory of both motor function as well as life expectancy for these children. And it's been a blockbuster drug that many of you may have heard of. But the development for this drug took a decade and cost billions of dollars. And so what to do for the rare and ultra-rare disorders where the resources are not commensurate with those rare but not nearly as rare disorders. So I'll give you some examples. It looks like my slides have gotten a little bit distorted. But I think you can see here, this is a example of a single patient named Mila. She was diagnosed with Batten's disease, which is a disorder of progressive motor dysfunction, seizures, visual loss, and eventual death. And so her family approached Dr. Timothy Yu at Boston Children's Hospital, and he recognized the urgency of the situation and was able to develop a personalized, individualized antisense oligonucleotide just for her, called Millicin after her name. And he was able to do this in a year, which is tremendously remarkable, and took a lot of cooperation of the FDA in modifying some of their processes for drug approval. So Millicin was given to Mila, and she did have improvement of her seizures, and she did eventually pass away, so it was not completely effective. But nevertheless, this study and this development of this drug for Mila really transformed the landscape and opened up the doors for similar development of similar drugs for other patients with rare disorders. So there are a few examples here of some individualized treatments that have been developed and are underway. There are many, many more in process. And Ionis, the company that developed the nusinersen for SMA, has spun off a nonprofit foundation called NLORM, whose mission is to support the development of antisense oligonucleotides for individuals with rare disorders. And they've treated a number of patients thus far. There are a number of treatments ongoing. And it was with that hope that in 2021 we applied to NLORM for Levi with the hope that they would be able to develop an individualized, personalized medication for him and his mutation. Their process is ongoing, and the red arrow indicates where we are in that process. And as you can see, the process is long, but they are trying to develop a personalized ASO for Levi. The biology of Levi's mutation is complex, but at least in part, there is data to support that his particular mutation can impact and negatively impact the function of the normal GNAO1. And so with that in mind, the goal of our therapy is to develop an antisense oligonucleotide that knocks down or reduces the abnormal protein and allows the protein, the normal allele, to function on its own. And so while NLORM is doing their drug development, we are trying to prepare for how we are going to trial this medication for Levi. And this is where the Bow Foundation has been instrumental in supporting our efforts. And so, how do we know that a drug is working? And I think you've already heard from a lot of people today about defining outcomes and measures, and that's what we've really been struggling with on our side, not with GNAO1 as a whole, but with an individual patient who is quite unique, which provides even its own challenges. Knowledge of the natural history is key, and that's why the studies that are ongoing regarding natural history are so important, because they allow us to compare what we might expect to what actually happens. But as you've also heard, those studies are at their beginning, and in addition, the changes that occur are not don't take place quickly, and so are not necessarily suitable for measurements in a quick trial, like we are going a trial where we have to determine effectiveness quickly. So we needed to think a little bit harder about what measures we would use. Are the patient measures observations the most important, or the physician measures, and what can we find that's quantifiable and easy to measure. And I've been really thrilled to hear this morning the work that everyone else has been doing, because I'm hoping some of you can help in what we've been doing and trying to figure out for Levi. And so we have been trying to find measures that are specific to Levi's needs, measurable, realistically obtainable in a short period of time, relevant to Levi and his family, and things that happen quickly. We've been using a mixture of physician and patient rating scales, some of the things that you've mentioned here, and are aware of many of the challenges of the existing scales, and have been focusing on the cognitive and motor realms. Levi's family has highlighted that communication and motor function are the top priorities. And we've been measuring baseline while we wait for the drug. My colleague, Dr. Natasha Ochkumov, who's been helping us with the cognitive assessments, which are a challenge in this group, was not able to be here today, but she did record a few slides, so I'm going to just let those slides run, where she highlights our evaluations of various assessments in other children, not Levi, but other children who are matched cognitively and motorically, so that we can see what, if any, measures we can find that would be useful. Are we able to get volume? Oh, maybe it's going to go.
Hello, I
There
am
we go.
Dr. Natasha
Okay.
Ochkumov, professor in the UC San Diego Department of Pediatrics. The challenges we face in direct assessment of neurocognitive abilities in this patient population include limited upper body coordination and limited verbal communication skills. A traditional IQ test with children over the age of 4 or 5 is not appropriate for individuals with intellectual disability of this type because their scores will be at the floor. The Bailey scales provide information about early cognitive, language, and motor skill development. With children outside the age range of the Bailey, growth scale values can be used to track growth over time. There's been interest in using the iPad-based NIH toolbox to assess individuals with intellectual disability. We decided to conduct a small pilot study to assess feasibility of these measures. Note that only the picture vocabulary test proved to be most useful given the limitations of the patients that we tested. Dr. Aaron Catt at Northwestern University also generously shared the concept formation test that he and his colleagues are developing within the toolbox framework. I tested 3 volunteers, and here's what we found. The first patient is a 7-year-old boy with intellectual disability and cerebral palsy. He responded using the touch screen. On the NIH Toolbox Picture Vocabulary Test, the child is presented with 4 pictures. A word is said, and you point to the picture that matches the word. His score is at the 5th percentile for age. On the concept formation test, he obtained 94% correct. The second patient is a 23-year-old boy with cerebral palsy and intellectual disability. He is not able to use the touch screen, but he could verbalize his choice. On picture vocabulary, his score fell at the 1st percentile for age. On the concept formation test, he obtained 71 out of 71 correct, 100% correct. The third patient is an 8-year-old girl with cerebral palsy and intellectual disability. She was not able to use the touchscreen or verbalize her responses. However, I pointed to each choice and she said yes, or she shook her head no, indicating her response. On picture vocabulary, her score fell in the average range, 58th percentile. And on concept formation, she obtained 68 out of 70 correct, 97% correct. It will be helpful to use change-sensitive scores and growth scale values to assess effects pre- and post-treatment. Unfortunately, we found ceiling effects using the concept formation test, and there are continued response modality concerns, given the limitations that we face, researchers are developing tasks that may rely on eye movements for responses rather than using the touch screen.
Similar to our cognitive assessments, we've been working with our rehab medicine team to evaluate motor function, in addition to the standardized scales that you've heard a bit about. And you've heard some of the challenges, so we wanted to examine some unique ways of measuring motor tone. Levi and a number of other children have a mixture of dystonia and increased tone in the limbs. And our group has been using a sensor glove to evaluate tone in their CP population, and we decided to try this with Levi. The way this is utilized is the examiner wears the glove with the sensors and repeatedly flexes and extends each limb, and the torque is measured and plotted over time. Levi's undergone a number of assessments so far, and I just show a couple of examples here to show change from one visit to the other. So ideally, we're looking for a stable baseline to establish a stable baseline before treatment. And as you can see, unfortunately, we're having some difficulty with stability, with some limbs showing increased tone, others decreased tone, and others staying about the same. Now, our measurements were confounded a bit by some treatments, surgeries that had to occur. As someone asked about Botox, that also needed to be given in between. And so we're hoping now, with a period of stability, to repeat these measurements more frequently to see if we can establish a stable baseline. We did also use a video-based assessment of joint movements to see if we could quantify a particular task, a reaching task. We were unable, unfortunately, to find a task that was doable and reproducible, and so we are still working on this. So as n-Lorem continues to work to develop the drug, we remain hopeful that they will be successful and will be able to identify a compound that Levi can trial. There's no guarantee that they will, and we are hopeful that even if they don't, that our attempts to develop some motor assessments and cognitive assessments applicable to either Levi or other GNAO1 patients will be helpful in the future. So we continue to work patient by patient to try to find effective therapies. And I'd like to thank the Bow Foundation, my collaborators who are helping us with our assessments, of course, n-Lorem for developing the drug, and mostly Levi and his family for taking this leap of faith and trusting us to work on the cutting edge of science to try to develop a therapy. So thank you very much.